Well, the responses and the durability with axi-cel CAR T-cell therapies are highly encouraging, with about 40% having long-term durability. About 60% of these patients relapse or progress after axi-cel. So we try to understand what are the potential mechanisms of resistance to axi-cel therapy. So we performed based analysis on the baseline tumor biopsies as well as tumor biopsies at progression. And we looked at by immunohistochemistry as well as RNA-seq analysis. By immunohistochemistry, we find that the baseline CD19 IHC score varied anywhere from 0 to 300 in these patients, but we found consistent effect with axi-cel regardless of the baseline CD19 H score. The CR rate and the PR and the durability of the responses were no different between patients who had lower scores versus higher scores. And then we also looked at biopsies obtained at the time of progression to understand why did these patients relapse after initial response. And we find that about 30% of these patients have loss of CD19 at the time of progression. And this loss of CD19 appears to be mediated either because of downregulation of the CD19 gene or because of alternative splicing of the CD19 gene, both of which can potentially cause antigen escape and progression of after axi-cel. Now, we also try to see whether these tumors that are relapsing after axi-cel can preserve other expression of other B-cell lineage markers. So we looked at CD20 expression, CD22, as well as CD79A expression, all of which are B-cell lineage markers. And we found that these tumors at progression, all of them retain CD20, 22, and CD79A expression. So altogether, I think this data suggests that one of the major mechanisms of resistance to axi-cel is CD19 loss, but this can potentially be overcome by targeting other B-cell lineage markers with CAR T-cell therapy, including CD20, CD22, and CD79A. So there are strategies now that are going to be entering into clinical trial in the near future to target CD20 CARs or use a bispecific CAR targeting both CD19 and CD20 or CD19 and CD22, which look very promising in the preclinical studies.